Alright. Michael Goga.
Here.
Kaylin?
Here.
Sarah?
Here.
Anna? Anna?
Bruh.
Is she here?
She was in clinical. Yeah, she was.
She's here. I think her mic is. Anna's here.
She might have went home.
I saw her before. Where are you? Anna? Yeah? Where are you? Oh, there we go. Oh, I see her. Hi, Anna. Okay, I see you.
God.
How do I make it not full screen anymore? Alright. Who am I on? Okay. Anna, Ro.
Here.
Charlie.
Here.
Omar.
Here.
Anthony.
Anthony's is, I swear he's here, but like,
He's here, but his mic doesn't work.
Yeah, because we're working during clinical. Yeah, his audio is not working.
Do I see him anywhere? Oh, and his video doesn't work either.
The video's working last year. I don't know. Yeah.
Anthony.
I think it was basic. I spelled how my
You can type, right? You should be able to type, no? You spilled Mountain Dew on your what?
Laptop. On my laptop.
Oh gosh.
No, I think it was just all that stuff.
Juliana?
Here.
George?
Here.
Good. Arya?
Here.
Good. Arna?
Here.
Jasmine?
Here.
Hannah?
Here.
Good. Mary?
Here.
And Olivia?
Here.
Okay. So what we're gonna do is we're gonna go over I think that what I'd like to do, sorry. You guys can hear me and see me and everything?
Yeah.
Okay. So what I think I'd like to do is go over so we'll go over the PowerPoint. Today we're gonna just talk about this one experiment called Messelson install. And then you guys noticed that on Classroom I posted the worksheet for it.
I thought
I
it.
posted this extra worksheet that
I tried printing it out, but it wouldn't print for me.
what the worksheet
Yeah.
it wouldn't print?
No, it wouldn't print. Well, it was just my printer issues, but like I couldn't get it to print. But I have it like pulled up.
Okay, so I'd like you guys to do those like either while we're talking or after, or maybe both, just to kind of like reinforce it, the material and stuff. And then there's another packet though, separate. So the guided notes I want you to do and then upload it back to me, like, and submit it right on Classroom. But the other packet I want you to do like separately, because it's like a little, they're both like review of the same concept, but I want to make sure you can like that you fully understand it, right? And so another thing, so I'm gonna go through the PowerPoint, and you guys, it's good that you guys all already muted all your things. But if you have a question, I mean, it's gonna be in full screen mode, so I won't be able to see like if you type it. So you can just come off of mute if you have a question, and you can like ask. All right? Good. Good. Okay. All right. I can't hear anyone right now, so that's fine. Okay. So are you ready to get started? Yes.
Yes.
Yes. Okay. So you have your notes out, and you have the guided notes out too, right? Okay. This is so awkward because like there's no sort of like response at all. So I really feel like I'm just talking to myself. But all right. So we're gonna try to make this as painless as possible. But this experiment is kind of confusing. So we'll go pretty slowly with it, all right? So let's see. And it's not that much. So I'm gonna present the slides that I have to you first, and then you guys can talk about it a little and ask questions. And if you still don't understand it, I can do something on the board here. Do you guys know which room I'm in? Can you tell? The dynamics.
Yeah.
Right?
Dynamics.
Yeah, because someone else is using our bio room, which is fine because we don't really need anything particular in there. Okay. So yeah, so I'm gonna go over it like once through the slides, and then maybe I'll illustrate it again on the board just to make it a little more clear. And then once we're done with that, we can disconnect, and then you guys can just work on the packets and then submit them You don't have to submit them by the end of the hour, but you should submit them by tomorrow, like the start of class. All right? We're good? Okay, so now I need to figure out how to do this. Someone please tell me like right away if you can't see what I'm talking about. You know, if you can't see the slides, because it's kind of like technically difficult to do all this stuff. So
you just have to like click on the 3 dots that are in like the top right corner, and then it should say like screen share.
yeah, I know, but then sometimes like it comes out. Even if you do that, sometimes it comes out like Alright. That's fun, right?
Wow.
Alright. Alright, so you see this, right?
Yeah.
Okay. You see this, right? I'm gonna hide this part right there. Oh no.
Yeah, I see. Oh.
That's better, right? Are we good? Okay, so I'm gonna start. So we're gonna talk about an experiment, right? Another experiment that's really important into our like current understanding of DNA, which is called. It's performed by these 2 people, Meselson and Stahl, and then some people have actually like called this experiment the most beautiful experiment in biology, which is kind of like a huge statement to say, right? But it is really, really, really cool, and I hope that you guys can appreciate that and understand exactly like what it's doing. Because obviously, if you can't understand it, you might not appreciate why it's so beautiful. But anyway, okay. So you guys know the 2 people who arrived at the structure of DNA were Watson and Crick, right? You guys are familiar with them? And so at the end of their paper, the only thing that they wrote about was like the structure of DNA, that it was a double helix, right? And that the base pairs were, matching up inside the structure of the molecule. But they didn't really talk about how it's actually replicated, because that's one of the functions of DNA, right? It stores information, and then it copies the information, and then it transmits the information, right? So anyway, so this is a quote from the actual paper. It says, it has not escaped our notice that the specific pairing we have postulated, so the pairing between the bases they mean, right, immediately suggests a possible copying mechanism for the genetic material. So basically, in other words, do you see the strand of DNA here, right? Basically, each strand serves as a template, and then based on that strand, you can figure out what the other strand is by just matching up the base pairs, right? And that's what they were trying to say. They were trying to say like that. That's how DNA is going to replicate itself. It's just going to base, and I don't mean to, the pun, base, but it's going to create the new strand based on the bases that are on one end, right? So that's what they were trying to say. That's what they were trying to predict. Okay? Is everyone with me? Yes? Good? Yes? No?
Yes.
Yes. Okay, good. You guys can unmute your phones, your mics every once in a while, just to contribute, an answer if you'd like. You don't have to be like totally muted, so don't feel you have to do that, but you can if you want. Okay, so I'm gonna move on, right? So, do you guys know when DNA replication occurs? Do you guys know anything about the cell cycle? No? Pardon me?
During mitosis?
Actually, that's when the cell divides, but that's not when replication occurs. But that's a common misconception, so I'm glad you said that. But so basically, there's this whole thing that all cells go through, and it's called the cell cycle, right? Which is the majority of the time that it's in? Is it?
Interphase.
So it's mainly interphase most of its time, right? Mitosis, which is pretty much cell division, where it actually splits into 2 cells, is only a small fraction of the whole life cycle of a cell, right? So actually when replication itself occurs, it occurs during the S phase of interphase. So basically right in the middle. So here's all of interphase, and then in the middle of interphase there's this period called the S phase, which stands for synthesis, and that's when the DNA is actually being duplicated, right? So from then on, this whole second half of the cell cycle, of the second half of interphase, you're all dealing with double amounts of DNA the whole time, that whole second time, right? The whole second half, right? So it happens during SKs and interphase, and it does not happen during mitosis. So make sure that you differentiate cell mitosis, which is just the division of it, versus like when it's actually replicated. Okay, that's really important. I'm gonna move on. Everyone, if no one objects, yes. Okay, so basically, they didn't know how DNA replication occurs, right? But they had three possible candidates, right? So the first one, right? And do you guys see that original DNA strand? So the original DNA strand is blue, right? And we're trying to figure out how it's going to turn into two new DNA strands, right? So can you guys take a look at this, right? In the conservative model, what happens to the original strands of the DNA? They become, do you see how they kind of come over here as is? So they basically remain totally intact and paired with each other, but then this new one, which is represented by the yellow, that is just synthesized based on the original one, but the original two strands stay together, and then there's two new strands formed, right? Do you guys see that? Yes? Good? Yes, no? Yes. Okay. And then after that, let's look at the next sort of generation. If they were to divide again, so each of these strands is now going to divide, right? Or replicate, right? Sorry, not divide, replicate. So do you see how this blue one right here, the blue one that was made, that was like the original parent one that kind of came down here? That one again will remain intact by itself, but form another one over here, a yellow one based on the original, right? So they're identical, but the actual two original strands stayed together, and then they just made two new ones out of new materials, right? Are you guys with me? And then this new one that was made based on that first one, right? It's the same as the original strand, but it's made of new materials, right? So that one will then go intact over to this one, this area right here. You see, that's the one that this becomes that right here, and it makes a new strand entirely, right? But so that's the conservative model, right? So conserving meaning that the original two strands stay together in their original form, and then you just make a copy, like a photocopy of it, right? All right. The next model was called the semi-conservative model, and can you guys see what's happening differently here? So what happened to this original double-stranded molecule of DNA? Anyone? Harsh, is that you? So what's happening here? Can someone tell me?
They basically alternate DNA, like the new strain and the same one from before, and they create two different strands.
Exactly. So the original two strands, they actually split up, and then they pair with a whole new strand here, right, to form two strands that are like half new, half old, right? Do you guys see how that worked? So there's this whole blue strand here, and then there's this whole other blue strand here, and then the whole yellow strand was made, and then the whole yellow strand was made here, right? And then to make the next generation, again, so this blue strand will separate with the yellow strand, so the yellow strand will come over here, and the blue strand will come over here, right? And then the even new materials, new yellow materials, will come in and pair up with the original strand, but then also pair up with this one. Do you guys see how that's happening? So how many original strands were there? There are two original ones, but those two original ones don't stay together in the later generations. They actually they just become like the strand itself stays together, right? But the two strands are separated from each other. Okay, so that was the semi-conservative model. And then there was a third model which they thought might happen, which is called the dispersive model. And in this case, I don't know if you guys can see really what happened here, but basically this original one, the whole original strand and this whole original strand don't stay together. Basically, you like chop up all this, right? Chop it up into like different little random pieces, and then in between you intersperse the new pieces of newly synthesized DNA in between. Right? Do you guys see how that happened? Right? And then in the subsequent generation, it's the same thing. You split it, you're chopping them all up, and then sort of like redistributing them like in sort of a random fashion, but so that like all of them are identical. But then the original parent DNA it's all interspersed among all of the offspring DNA every time. Okay? Does anyone have any questions about this so far? No? And are you guys following along in the packet with, are you guys following along in the packet, like, of the, like, sort of guided questions that I gave you, or?
Yeah.
Am I answering all the questions there?
I'm following along, but how are we going to be, like, tested on this?
I honestly have no idea. So, one of the things that you are going to do is, so, like, and I have to sort of figure out how we're going to do this, too, because originally, like, one of the assessments for this unit is that you're supposed to make a video of DNA replication, and it's a group project, and I think that it's like too much to ask of like maybe one student to do, like each student to do on their own, but I don't know how you guys are going to collaborate. Are there like things online that you can use to collaborate and make a joint video?
Well, like, I don't, kind of depends on, like, I don't know,
We
actually.
could always, like, all pick parts of the video and then, like, put them together in, like, iMovie or something.
Yeah.
Yeah.
It'd just be hard to
Yeah,
get
like-
everyone out.
Or, well,
It
everyone's,
definitely like require a lot of coordination, and like because otherwise, like you want it to be consistent throughout the video. Maybe you guys could do like a slides, like a joint, like a shared slides thing, animation thing. I don't know. We can sort of talk about like what resources there are available, but you guys would rather do a group project than an individual project, right?
yes.
Okay. I think that definitely like divides the labor up a little better, even though it does require more coordination. Okay. All right, good. All right, so you guys are all set with these 3 different models, right? Does anyone have like a sort of a hunch as to which of the models is correct? Of how DNA replicates itself?
Yeah, dispersive.
Okay, some of you guys think dispersive. Any semi-conservative?
Semi-conservative.
Who is that?
Omar.
Omar. All right.
I would go with semi-conservative.
Who says conservative? Lidu?
I agree.
No, I say semi-conservative.
You say
I
semi-conservative?
agree with you.
All right. All right. Well, we'll see. Let's see what happens. And let's see how this experiment. Okay.
I also.
Who is that?
Thank you, Ari.
Aria.
Yeah.
Oh, Aria, you're here.
Hello.
Wait, you didn't go. I thought you, for some reason, I thought you weren't going to be here. But I guess now that it's remote,
No,
did your trip get canceled? Oh, it got canceled. Oh no. Okay. Well, good to have you here at least. That's good. All right. I didn't even think about that when I was taking attendance. But yeah. Okay. All right. So how are we gonna like just determine which of the 3 methods it is? The Meselson install experiment is the one that included that, right? And there are a few different things that we have to understand before we can actually understand how this experiment worked. So first of all, do you guys remember what a centrifuge does? Anyone?
Doesn't it like spin the things around and like puts the denser things on the bottom?
Exactly, perfect. So it spins things around, and it was used in which experiment that we already talked about?
Hershey Chase.
Hershey
Wait, is that
Chase, very nice, good. So in Hershey Chase, they used a centrifuge to separate the like heavier cells, right, bacterial cells, from the lighter bacteriophages, right? And in the Hershey Chase experiment, it basically just formed 2 separate layers. There was the pellet, which was the denser part, and then the supernatant, which was like the liquidy part, right? But not all centrifuges necessarily have like a solid part and then a liquid part. Some of them have more of a gradient, where like things that are heavier, again, will they be lower or higher in the tube? Things that are denser or heavier. They're going to be lower, right? Exactly. So basically, a centrifuge, it separates some substances based on their density, right? The heavier ones are going to form, in this case, it's not like pellet versus supernatant, it's just like bands, where all those those things of that density are going to sort of like congregate or like be like pushed to when like by gravity, right? So the heavier ones are going to be lower, and then the lighter bands are going to be higher, right? So it's going to be like a banding pattern. So if I see like this, you know, this tube, and there's like bands in the bottom and then bands in the top, I can assume that all the molecules that of the lower density of the higher density form the bands on the lower part, right? Okay, pretty easy, right? Do you guys remember what an isotope is?
Atoms of this.
Sorry, you got cut off, George. Say it again.
Atoms are an element with a different amount of neutrons.
Exactly. So they're going to be different in the amount of neutrons, and then what other number is that going to affect?
Mass number.
It's going to affect their mass number, and so basically, the ones with more neutrons are going to have a higher mass number, so they're going to be heavier, right? Okay. So yeah, it's a different version of an element with a different number of neutrons, and therefore a different mass number. Okay. And then how do isotopes differ between themselves, like in terms of activity? Do they have different chemical bonding patterns than like other isotopes? They have the same ones. Why? Because they have the same number of what?
Electrons.
Electrons, and electrons are the ones that are involved in the bonding process, right? So the isotope, the only difference is that the isotopes have are sometimes that some are radioactive and some aren't. So that makes them really helpful in like being able to track certain like molecules. If you want to know where all these like molecules are going, you can use a radioactive isotope and then sort of detect for radioactivity, and then you'll know where those things went, right? But the chemical behavior of isotopes is more or less the same for our intensive purposes, right? So in the Hershey-Chase experiment, they used what kind of isotopes? Like the isotopes differed from their normal isotopes in what property?
Radioactive.
Exactly. The ones that were used were radioactive, so they could detect where those molecules were going, right? So in the Michelson-Stahl experiment, they do not use radioactive isotopes. They're just using isotopes with different masses, so that they can determine the difference of two different isotopes based on where they form in a centrifuge tube, right? Do you guys kind of understand what that means? Yes? All right. I'm going to take that as a yes. Okay. All right, so let's, so again, yeah, how do they differ? By radioactivity or by mass? But not all of them are radioactive, and but all of them will have a different mass, right? Okay, so let's move on. So the first part of the experiment, basically they took these E. coli cells. What kind of organism is E. coli? Do you guys know?
Virus.
It's not a virus. It's a bacteria. Okay? So they took these bacterial cells and they grew them in an environment where there was only nitrogen 15. Okay? So, and we know that like in DNA they have nitrogenous bases, right? So all the DNA that's grown in that environment will have what kind of nitrogen in it? If it's only exposed to nitrogen 15, what kind of nitrogen will all the DNA have in it?
Nitrogen 15.
It will all have nitrogen 15, right? All the E. coli cells, right? So basically here are the cells, the blue ones that are grown on the blue medium of nitrogen 15. And we'll call those generation 0. So that's like the original parent generation. Are we good? Yeah? Okay. And this phase of the experiment, they just called the pulse phase. So this is the pulse. They're growing the parent generation on this heavier nitrogen medium. Okay? So N15 is like heavy nitrogen. That's the nickname for it. Okay? So all the cells here, if we were to purify these cells, so take out all the other stuff, like take out the cell, the cytoplasm and all this stuff, and isolate the DNA, right? And if we were to then centrifuge those, right, what would it look like? What kind of all the cells are pretty much the same species and they have the same amount of DNA. So if we isolate the DNA from those cells and we centrifuge them, basically they'll form a single band, because they all have the same mass, right? But it's going to be at nitrogen, at like this random lower point that we're going to call nitrogen 15. We're going to label that as nitrogen 15. Are you guys okay with that?
Yeah.
Yeah? Okay. All right. So then, after that, so remember this band pattern. There's going to be one at nitrogen 15. All right? So then what they did was, all right, so then they transferred those G0 cells, the ones that were grown on nitrogen 15, so they band pattern at nitrogen 15, right? And they transferred them to a nitrogen 14 environment. And then they let them divide once. Okay, so here are those original cells, and then they let them replicate one time, right? So now they're going to have like children, basically. They have these other cells that are like the offspring of the first generation, but when they're making their new DNA, they only have access to what kind of nitrogen? 14, right? So they're going to add in so they can't add in any more nitrogen 15. So the only thing they can add in to make their new strands of DNA is nitrogen 14, right? Okay, so this once they move them to this other environment, that's called the chase phase of the experiment. So the whole experiment is called pulse chase, which means the pulse phase is the nitrogen 15 environment, and then the chase phase is the nitrogen 14 environment, right? So they let them divide, or they let them replicate for one generation, and then what do you think their results were? Right? If you purify and centrifuge the new cells, what do you think we're going to get? What kind of banding pattern do you think we're going to get? And I want you guys to think really carefully about this, and then we'll go through the possibilities, all right? Are you guys with me? Does anyone have any questions? Questions? No. No. Okay. All right, so let's look at all the different possibilities, right? So this was the parent one was G0, right? G1 is the first generation after they were allowed to replicate in the new environment, and G2 is the second generation after they were allowed to replicate twice, right? You guys kind of understand this? Yes? All right. So if it were so first of all, the parent generation, what nitrogen were they made of again?
15.
Nitrogen 15.
Nitrogen 15. So if you centrifuge the DNA from those cells, what kind of band would they make? A band where? At nitrogen 15, right? No like descent there, right? Okay, let's look at the semi-conservative model, right? So what kind of band pattern would you expect if you have molecules that are half one strand of nitrogen 15 and half one strand of nitrogen 14?
It would be in the middle of where nitrogen 15 and nitrogen 14 are located.
Exactly. Who said that?
Lidu.
Lidu. Very nice. Good. So it's going to be for these two. And how many bands will we have? One or two? What's the relationship between these two molecules here? They're identical, right? And they're going to be identical in mass because they both are one strand of old and one strand of new, right? So in this one, we're going to expect what kind of band pattern? A band pattern at like what? N14.5, let's say, right? Are you guys all with me for that? What about for this conservative pattern? What would we expect the band pattern to look like if we have one molecule that was nitrogen 15 and stayed nitrogen 15 entirely, and then the whole new molecule which is made entirely of nitrogen 14? What kind of bands would we have? How many different bands would we have? All these molecules are going to centrifuge where? At nitrogen 15, right where their parent, because right where it did before, right? Where are all these bands? Where are all these going to separate? At nitrogen
14.
14, right? Because they're lighter. So we're going to expect two bands here. One's going to be at nitrogen 15 and one's going to be at nitrogen 14. Are you guys with me there? Yes? Okay. And then dispersive, what would we expect here? So again, it's the same, it's the parent DNA, and it's just being chopped up, and then interspersed with like little inserts of new DNA. But what's the relationship between these two to each other? Are they going to have different masses or the same mass? They're going to have the same mass, right? So how many bands does that mean they'll have? Was that Addie? How many bands will there be then? Just one, right? And it will be where?
14.5.
It will be of an intermediate because it's mixed between 14 and 15. So again, this one will also be at like 14.5, right? So it's going to be, this one's going to look like that. You guys all agree, right? This is this band down here. This represents nitrogen 15, and this represents nitrogen 14, right? This one would have two bands, right? It would have one band at 15 and one band at 14. You guys all agree with that? All right. And then this one is going to have, again, the same pattern as this one because they're both like mixed molecules of both 15 and 14, right? Does anyone have any questions so far? No? Okay.
Are you
So
able
no?
to post the notes afterwards? Like the PowerPoint?
Yes, of course. Yeah.
Thank you.
Okay. And I also have a worksheet. Like, so I want you guys to do, so the worksheet, my worksheet, like my homemade one, I want you guys to answer those questions and then submit that back to me on Classroom. But then the other packet, I also want you to do, like, just on your own. And I guess, I don't know how I'm going to check that because you can't, like, edit that document itself. Maybe, like, take a picture of it and then send that back. I think that's maybe what we'll have to do because I want you to do that one too. It's going to, you might feel like, oh my gosh, this is like a lot of repetition. But for those of you who don't really fully understand it, I'm trying to give you different ways to be able to understand it, okay?
I printed it out.
Or no?
Can I take a picture of it and like post the picture that I printed out? Like,
For which one? For the notes?
For for both of them.
Yeah, if you guys do that, that's fine too. But then just, just do whatever, if you want to take a picture and then submit it, or if you want to like actually print it, like type it out and then submit it, either way is okay. But make sure you do both of them, right? Alright. Okay, so let's, I'm just going to call in random people now. So, alright, so going back to the semi-conservative model, right? So after one generation, we noticed that there would be one band. You guys all agree with that, right? And it would be an intermediate band between 14 and 15, right? What about after the second round of replication? And replication takes about 20 minutes in E. coli. So after the second round of replication, remember these two strands are separating from each other, and then we're adding new DNA to be complementary to the original strand here. And then this was that second strand made in the second round, but then it adds new nitrogen 14 DNA to that one. So what will this band pattern here, how many different types of molecules do we have here? Charlie. How many different types of molecules do we have in this one?
2.
We have 2 different types. So how many bands will we have in our centrifuge pattern?
1.
We have so each one will form a different band at that whatever that respective like weight is. So these 2 like mixed ones are going to form one band at one place, and these 2 lighter molecules are going to form another band at another place. So basically we'll have 2 bands, right? Where will these 2 bands be located in the tube? Olivia.
The middle and the bottom one.
These 2 will be,
Or the top and the bottom.
yeah, these 2 will be located in the middle, and then these 2 will be located in the top, right?
Yeah.
Perfect. So you would expect for this one, they would be like, they would be bottom and then middle. Middle and then top, right? All right. Moving on to the conservative model. Let's see. How many different types of molecules do we have here? Arsh.
There will be two.
So we have two different types of molecules, one that look like this and one that look like this, right? So there'll be two bands. Where will the bands be?
An N15 and N14.
Perfect, right? What if we let it divide for another generation in N14? What will it still look like?
So we'll have one band at N15 and one at N14.
Yeah, so forever, it's always going to be one band at N15 and one band at N14 for this model, right? All right, and what about for this last model over here? So remember, we're just chopping them up and then we're kind of redispersing them, right? So how many different types of molecules in the third generation here do we have for this one? Or not? How many different types of molecules do we have?
One.
We have one, so how many bands are we going to have?
One.
One, and where will that band approximately be?
Like near the middle.
Near the middle, but for each subsequent generation, we're adding more and more what to this? Yellow or blue?
Max. More, yellow.
Yeah, more and more yellow. So that band will always form one single band, but that band, the more and more generations we go through, what's going to happen to the band? It's going to start moving slightly up or down?
It's gonna start moving up.
Exactly. Why? Because we're getting more or less dense?
Less dense.
Less dense, because we're adding more of the lighter nitrogen inside of that. Good. Okay, so in the real experiment, what happened was after the first generation, they saw one band, and it was at intermediate level density, right? So which model can we eliminate after that first round when they saw just one band? Let me choose someone. Mary, which model can we eliminate if we saw just one band?
The conservative one.
Good. So that one clearly after the first generation was gone because they knew that it wasn't. It was one band, not two, right? And then, so how are we going to distinguish between whether it's dispersive or semi-conservative? Anthony, where do we have to look now? Oh, he can't talk. I forgot. Sorry. Let's see. Caitlin? Hello? Are you guys all there still? Are you guys all there? What happened? Did it hang up?
Yeah.
Oh. Are you guys still there? Can you see me?
Yep.
You can see my screen still?
Yeah.
Yeah.
Alright. Alright. So I'm just going to continue. Is that alright?
Yep.
Okay. So, oh gosh. Okay. So, did it do it again? Did it do it again?
I don't know, the screen's not weird right now. Now I see your face.
Oh, really?
Now I see George.
You guys hear the ringing?
Yeah. We're not getting a call.
Who's getting a call? Am I getting another call?
Probably. I don't know, but.
Really popular in this little house. I can't even see my. Oh, here. You guys can't see my. You can't see my. Oh, I was getting another call. That's why.
Yeah.
Hello?
Yeah.
Okay, sorry, you're still here. All right, actually, it was like another teacher calling me to try to test something on like this thing. I don't know, but anyway, I just told her to stop calling. All right, let's go back. All right. Let's see, 3 dots, share screen. Your entire screen, yes. Okay, share, and then. Okay, here we go. We're almost done, guys. I promise. All right, so I'm hiding this. You can just see this craziness again, right? Oh, there we go. Are we back? We're good?
Yes.
Okay. So
Yes.
who was I calling on?
On Michael?
Okay, Michael. So, can you just summarize again? What would I expect? So after this, so the real way that I'm going to distinguish between dispersive and semi-conservative is if I go to the second generation. So I'm going to let them replicate one more generation in nitrogen 14, which is the light nitrogen, right? And then I'll see different band patterns based on each of these, right? We already ruled out the conservative, so we don't have to do that one. So for semi-conservative, what am I going to expect to see again?
For semi-conservative, you're gonna expect to see 2 yellow and then 2 of the mixed one.
Right, and then what would that band pattern look like in the tube, in the centrifuge tube?
I don't know.
So these mixed ones, these like half and half ones, those are gonna be of an intermediate density between 14 and 15. So they'll form one band, right, at 14.5. And
Yeah.
then these other yellow ones, those are light, right? So where will those form a band?
14?
At 14, right? So
Yeah.
it's like 2 bands, right? One at 14 and one at 14.5, right? Okay. And then this, sorry, the next one for dispersive, right? How many different types of molecules were there again? I think Arnav answered this before, but Hannah, how many different type of molecules are there here? Hannah? Are you muted? Are you there? Is anyone there?
Can I get off here? Answer the question. There, it's not talking.
She's not talking. All right, let's see. Juliana, how many different types of molecules are there here?
1.
There's one, right? So how many bands will we form?
1.
And then what will at what level will those bands form at?
Like intermediate.
The intermediate, right? So exactly. So it's gonna look like this, right?
And
And
so
so-
not letting her click on you.
Oh, it's not letting her click? Okay. Alright, that's alright. Okay. So then what do you think they saw? So after the first generation they saw 1 intermediate band, and after the second generation they saw 2 bands, 1 intermediate and then 1 at 14. Right? So that allowed them to conclude that which is the actual mode of DNA replication? The semi-conservative model, right? So that's like the takeaway message. But you guys, I do expect you to know what each of these like test tube, like centrifuge tubes are going to look like after each round for each model, right? So basically the takeaway message here is just that DNA replication is semi-conservative. Okay? Meaning that one strand of the original parent one is totally conserved, right? So basically it's made of 2 strands, right? And they separate from each other, and then the new DNA is like built onto that parent template, right? But the new DNA is coming from it's not like the parent one does not stay as it was originally. All right? Are you guys good? Yeah? All right, let me stop sharing my screen and let you all right, so you guys are all okay with this? You feel comfortable?
Yeah.
Yeah?
Pretty much.
Okay. So you guys were able to fill in all the questions in the packet that I gave you?
Mhm.
Yeah? Alright, so that extra primer, that should like serve as an extra review in case you weren't like totally comfortable with this. But you should recognize that the 2 original strands of parent DNA, instead of just photocopying themselves and then going off to one side by themselves, they're actually split. And part of them is actually used to make the next round of DNA. Right? So they're splitting. Yes. Who is it?
In the packet, for generation 0 and generation 1, it
Who
says
is
like,
it?
what are they?
Oh,
What's
there
it
you go.
referring to?
Say it one more time.
In packet, it says like, what was generation 0 and what was generation 1. Is that referring to like the phases?
So generation 0 was the group of cells that was grown on heavy nitrogen alone.
Oh, okay. So,
Right. So and you can also call that the parent generation, the original parent cells. And then generation 1 would be the first, like the offspring of the first round of replication in nitrogen 14. And then second generation, or the generation formed after the second replication in the nitrogen 14 environment. Okay? Are we good? Alright. You guys were great today. This is good. Omar, what are you wearing there? I can't hear you.
It's an old Halloween costume.
Lovely. Is that just for our benefit, or were you already wearing that?
I was wearing it like the entire day.
You were. Excellent. Well, that's an interesting choice. Arch has his hat on, so that's you guys all dressed up and ready for the day. That's good. Alright, so does everyone understand what happened or no? Can anyone can someone explain it? Can I have a volunteer to explain the whole experiment?
I will try.
I will.
Who said I'll try?
Alright.
Who?
Alright.
Raise your hand. I don't see you. Arsh? Alright, Arsh is gonna do it. Everyone, let's listen to him and let's make him the biggest one on our screens.
It's gonna go very badly, but basically what happened was, so everyone was like, this DNA stuff, it definitely replicates itself, and everyone's like, how do we do that? So then these two guys, they were like, so why don't we test it by taking these these E. coli bacteria, and we'll make them breed and see what happens, or make them reproduce. So then eventually, what they did was they took heavy nitrogen, which is N15, and they breeded a bunch. They bred a bunch of them in just pure N15, and that and when they were able to isolate the DNA and they centrifuged it, they were able to see the results of the different of what happened with the bands of the DNA, and depending on the density of it, they were able to test or disprove the three theories of DNA replication.
Okay?
Basics, that's what it is, and the three bands, the three theories, there was conservative, semi-conservative, and the conservative, semi-conservative, and dispersive, and they were able to and just looking at it and by testing, they were able to figure out which one it was. So that's all I can offer.
Okay.
But,
Very nice.
thank you.
And so you guys, if I asked you what the banding pattern would look like after the first round of replication in N14, if it were conservative, you guys all know what that would look like, right? So if DNA replication had followed the conservative model, right? What would the banding pattern of the centrifuge have looked like after one generation in N14? Let me see who I'm going to row. Haven't heard from you yet today.
There would be one at N14 and then one at N15, so 2 bands.
Excellent, good. You guys all got that? You all knew that, right? Okay. Let me see. What would the banding pattern after 20 replications in N14 have been if the dispersive model were correct? Kaylin. To unmute it. Kaylin? Oh, there you go. So what would the banding pattern of the centrifuge have looked like if the correct model were dispersive and after 20 generations of replication in the dispersive in the N14? So after having switched from N15 to N14, then after letting it replicate 20 times in N14, what would the banding pattern look like for the dispersive model? You would have one band, and where would that band be? In the middle. But it would be progressively after each round, slowly moving up toward the 14, right? Excellent. All right. And then let's see. Hannah, can you unmute, or does it still not work? It doesn't work. Can you hear me though? You can hear me, right? I can see you, but I. Oh, what? Oh, you unmuted it. Okay. Oh, good. Okay. So what did the actual banding pattern look like after 2 rounds of replication in the N14 medium in the actual experiment? What did it look like after 2 rounds?
I don't like understand the question. Can you, like,
So in the actual experiment, after they took the bacteria from N15 to N14, and then they let it replicate for 2 generations, what did the banding pattern look like? Like, what did it actually look like for the semi-conservative model?
it was in the middle.
Well, how many bands were there?
There were 2.
There were 2 bands. One of them was in the middle, and where was the other band?
It was at N14.
N14, right? So there were 2 bands. So one of them was in the middle, and one was at N14. Because you're always going to have those 2 original parent strands, which are going to make half the molecule heavy, but then all the subsequent ones are all going to be at N14, right? All right, you guys are okay with this? Do you understand? If you have any questions, please email me, right? Please submit your work by, not the last minute, because then I can look at it a little bit and, sort of like gauge how you guys are doing with this, okay? Tomorrow we're going to do like the actual process of DNA replication with all the little enzymes involved. So it's going to be kind of technical tomorrow too. But again, I'll post the the notes packet and then any other like assignment that would be required. Okay. And if you guys have no idea what I'm talking about, please email me and then ask questions, or I can give you some more practice questions to answer if you want. Okay. This is an adventure. We're gonna have to just sort of adapt to this way of learning. But I don't know, you guys are up for it, right? And now it's like 12:30 and you have the rest of the day to yourselves. So are you guys quarantining yourselves? Are you staying in and not going out and being crazy? I hope.
Drones flying outside in the sky last night. There's like drones.
What? There are drones? There's also a curfew. Did you guys see that?
No.
Yeah.
So I hope you guys are being smart and enjoying the time, to really, to really enjoy, to relax, but also to, reflect on life and, be responsible citizens and not go out and crazy parties or anything, right? Okay, and I know it's going to be frustrating to learn like this, but I'm going to post the PowerPoints every day. I'm going to go through the PowerPoints with you, and then if you need extra, help, please just ask questions, email me. I'm still very available, so even though we're not in the physical space, I'm still at your disposal. So please let me know if you have any issues, okay? And in terms of assessments of how we're going to do that, we'll have to, we might have to come back and take a few in a row or something. I don't know, but we'll or maybe we'll figure out a way that you can do it from home, where you can kind of create some sort of answers or some sort of project to demonstrate your understanding, because that's the point, right? To show that you understand it. All right, guys. All right, so I'll see you. This worked really, really well. I'll see you guys same place, same time tomorrow. Okay. All right.
Have a good day.
Have a great day, guys. All right. Okay. Bye. See you. Bye.
will see you. Yeah, alright, thank you. Yeah, but I have a lot of homework. Why? I don't know, more than usual. Classes used to be an hour and a half, and now they're only an hour. And lunch was an hour, but now we have a break for 20 minutes. I don't know, what else are we going to do in 20 minutes? Yeah, go to the bathroom, come here, go take a shower. Who's going to take a shower, man? You like it? No, I don't like to see my teachers. You see them on the computer? Yeah. So we have to FaceTime them. On the phone? On my laptop. How come that's what they don't do that? They will, like, next week. They're using their spring break. How about you? Mine in April. Yeah, just, they're gonna use your spring break. Like, they're using the 3 weather days and his spring break. And they're using none of mine. Why you guys can't do? Why they? Because the teachers were trained on Friday. They didn't train them yet. They're probably doing it today. You hungry? Yeah. Why don't you eat something that we had last night? What did we have? Oh, I don't want that. What do you want? Something without vegetables. Mary, your body needs vegetable, man. I just to fight this virus going on, man. I don't know. Why don't you chicken nugget if you want? Are they going to test you? I don't know. Maybe for the moment. I have a body ache like this.